There are places where time loses its meaning, where water has been gone for decades, where temperature swings from lethal heat to absolute cold without warning, where radiation passes through matter unchecked, and vacuum strips away the last trace of air. In these spaces, biology does not struggle, simply fails. Cells collapse when water disappears. DNA fractures under radiation. Proteins unfold, never to recover. For almost all known life, these conditions mark a clean and final boundary. Beyond it, survival is not delayed, it is impossible. And yet, scattered across Earth's most ordinary surfaces, damp moss, thin films of freshwater, soil between roots, lives an animal that repeatedly crosses this boundary, not by resisting these extremes, but by stepping out of biology itself. It shuts down growth, metabolism, repair. It abandons movement, feeding, even aging, entering a state that resembles death more than life. Years later, sometimes decades, it resumes as if nothing happened. The survival is not passive luck. Each microscopic body is a carefully orchestrated collection of molecules designed by evolution to endure absence itself. The physical collapse of cells is prevented not by force, but by proteins that stiffen as water disappears, and by vitrification, a transformation that turns the interior of the body into glass. Motion, chemistry, even time itself nearly stop. Yet, when conditions improve, molecular scaffolding relaxes. Cells rehydrate, metabolism resumes. Life continues, seamless and unbroken. Today, we're diving into why tardigrades choose to endure into vacuum-silent extremes. Not as an accident of toughness, but as a biological strategy shaped by instability, absence, and repetition. Because the question is not how they survive space, radiation, or desiccation, but why evolution ever pushed life to survive by almost stopping altogether. Life rarely collapses in a single dramatic moment. More often, it erodes quietly. Water thins, then vanishes. Temperatures drift past tolerable limits. Exposure arrives not as catastrophe, but as routine. For tardigrades, these shifts are not rare extremes found only in distant environments. They are the background rhythm of daily existence. Tardigrades inhabit microworlds defined by instability. Moss cushions on rocks dry out repeatedly under sunlight. Thin films of freshwater evaporate within hours. Soil between plant roots cycles endlessly between saturation and drought. At this scale, there is no warning before conditions become lethal. Drying happens faster than movement. Freezing arrives before shelter can be found. Escape, the dominant survival strategy for larger organisms, is biologically unrealistic. Unlike animals that migrate, burrow, or thermoregulate, tardigrades are bound to surfaces measured in millimeters. Their environments oscillate rapidly between abundance and absence. A rainstorm may flood their world, triggering feeding and reproduction. Days later, evaporation strips away every molecule of free water. In winter, freezing locks remaining moisture into ice. In summer, heat accelerates desiccation. These cycles repeat not seasonally, but unpredictably. Under such conditions, resistance alone is insufficient. Evolving tolerance to heat or cold addresses only part of the threat. The more persistent danger is water loss, the removal of the medium that enables every biological reaction. Cells require water to maintain structure, transport molecules, and stabilize proteins. Without it, membranes collapse and chemistry ceases. For most organisms, dehydration is irreversible. Tardigrades do not avoid dehydration because they cannot. Instead, evolution pushes them toward a strategy that reframes survival itself. When environmental signals indicate drying, decreasing humidity, rising osmotic stress, tardigrades begin a controlled withdrawal. They retract their limbs. Their bodies contract into a compact, barrel-shaped form known as a ton. This shape minimizes surface area, slowing further water loss. But the transformation is not merely physical. Internally, metabolism declines toward near zero. Growth halts. Repair mechanisms pause. Cellular processes that normally depend on continuous chemical flow are shut down in sequence. What emerges is not adaptation to dryness, but a temporary suspension of biological time. This response is not triggered by space, radiation, or vacuum. It evolves because of ordinary environments that repeatedly erase the conditions required for life. Moss does not remain wet. Soil does not stay stable. Fresh water does not persist. Tardigrades survive extreme experiments because their everyday world already forces them to endure states that resemble biological absence. Crucially, this endurance is reversible. When water returns, tardigrades do not rebuild from scratch. They rehydrate. Metabolism resumes. Movement returns within hours. Reproduction follows. The cycle repeats without memory, without cumulative damage, at least until limits are reached. When a tardigrade contracts into its ton state, the most visible change is stillness. But the real transformation happens invisibly, at the molecular level, where biology faces a paradox. How to stop life's chemistry without letting it fall apart. In most organisms, dehydration is catastrophic because water is not simply a background condition, it is a structural component. Proteins depend on water molecules to maintain their shape. Membranes rely on hydration to remain flexible. DNA strands require aqueous environments to preserve stability. Remove water too quickly, and the architecture of the cell collapses inward on itself. Tardigrades confront this problem by not attempting to preserve normal biology. Instead, they replace it. As dehydration progresses, tardigrades synthesize large quantities of protective molecules that fundamentally alter the physical state of their cells. Among the most critical are specialized proteins that remain disordered in normal conditions but reorganize as water disappears. These proteins form a molecular scaffold, stabilizing cellular structures that would otherwise unravel. Rather than fighting dehydration, the cell reorganizes around its absence. At the same time, tardigrade cells undergo vitrification, a process where internal contents transition into a glass-like state. Unlike ice, which forms sharp crystals that puncture membranes and organelles, vitrified material is rigid but smooth, locking molecules in place without tearing them apart. Chemical reactions halt not because enzymes fail, but because motion itself becomes impossible. Metabolism drops to levels so low they approach the edge of measurement. Oxygen consumption becomes negligible. ATP production ceases. Repair processes stop mid-action. Yet crucially, this shutdown is orderly. Damage is minimized not by activity, but by immobility. This is cryptobiosis, not survival through resistance, but survival through suspension. In this state, tardigrades do not age in any meaningful biological sense. Aging depends on cumulative chemical damage, replication errors, and metabolic byproducts. When chemistry pauses, so does aging. A tardigrade that remains desiccated for 10 years does not emerge 10 years older. Time, biologically, has not passed. But cryptobiosis is not invulnerability. It is a narrow corridor between life and death. The molecular scaffolding that protects proteins can fail. Vitrification can fracture under extreme stress. Prolonged exposure increases the risk that damage accumulates beyond repair. The strategy works not because it is perfect, but because it is repeatable, effective enough, often enough to be favored by evolution. When water returns, the process reverses. Cells rehydrate, proteins regain mobility, metabolism resumes. The animal unfolds from its ton and begins moving within hours. No rebuilding, no regeneration, just continuation. This ability to suspend life without dismantling it is what allows tardigrades to face conditions that destroy nearly all other multicellular organisms. And it is precisely this trait that invites an obvious question. What happens when the environment removes not just water, but everything else as well? Vacuum, unlike drought or freezing, strips away atmosphere entirely. Pressure drops to near zero. Volatile molecules escape freely. Radiation arrives unfiltered. In theory, this should push cryptobiosis beyond its limits. To test that boundary, scientists have done what evolution never planned for. They remove tardigrades from Earth altogether. In low Earth orbit, outside the protective blanket of atmosphere and magnetosphere, organisms are exposed to vacuum and cosmic radiation simultaneously. Multiple spaceflight experiments sent tardigrades into orbit in dehydrated states, exposed directly to space for days and weeks at a time. some survived. When returned to Earth and rehydrated, a portion resumed movement and reproduction. Their cryptobiotic state had preserved enough cellular integrity to withstand vacuum exposure. The glass-like stabilization prevented rapid molecular loss. The absence of water limited radiation-driven chemical reactions. But survival was incomplete. DNA damage accumulated. Double-strand breaks increased. While some tardigrades repaired this damage upon rehydration, others failed. Reproductive success dropped. Survival varied by species and exposure conditions. Radiation proved the more decisive threat. Unlike dehydration, which tardigrades encounter routinely, cosmic radiation introduces damage that cryptobiosis cannot fully neutralize. Specialized DNA-associated proteins help shield genetic material, but they do not render it immune. Protection reduces damage. It does not eliminate it. These experiments reveal a critical point. Tardigrades are not adapted for space. They are adapted for unpredictability. Space is merely an extreme exaggeration of the instability they already endure on Earth. Vacuum survival is a byproduct, not a purpose. Endurance is not free. Evolution trades one advantage for another, and in tardigrades, the cost of cryptobiosis is complexity. Tardigrades are small, structurally simple animals. They lack advanced circulatory systems. Their nervous systems are minimal. They do not exhibit the behavioral flexibility seen in more complex organisms. These are not failures of evolution. They are consequences of a survival strategy centered on shutdown. Complex systems are harder to suspend safely. More interactions mean more opportunities for irreversible damage during dehydration. Simplicity increases the odds that vitrification and molecular stabilization succeed. Reproduction also reflects this trade-off. Tardigrades do not invest heavily in long developmental periods or elaborate parental strategies. They reproduce efficiently when conditions permit, then rely on cryptobiosis when conditions collapse. This approach works exceptionally well in environments defined by instability. It works less well in stable ecosystems where competition favors speed, specialization, and adaptability rather than patience. As a result, tardigrades persist quietly at ecological margins. They do not dominate food webs. They do not reshape environments. They endure. Their strategy forces biology to confront uncomfortable questions about the nature of life itself. If an organism can halt metabolism entirely and remain viable, where does life end and death begin? If time can be biologically paused, what does aging truly measure? Even tardigrades have limits. Extreme radiation overwhelms repair mechanisms. Prolonged exposure eventually degrades protective proteins. Cryptobiosis delays damage. It does not erase it. There are boundaries beyond which even suspended life cannot return. Tardigrades do not tell us where life thrives. They tell us where life barely holds together. They illuminate the boundary between biological function and biological absence, a boundary far thinner than it appears. At that boundary, life does not roar. It does not adapt rapidly or innovate endlessly. It becomes quiet. It becomes still. It waits. Their survival invites reflection on human assumptions about resilience. We tend to associate survival with activity, strength, or complexity. Tardigrades show that endurance, patience, and the ability to pause are equally powerful strategies, particularly where instability is the rule, not the exception. If life can stop without disappearing, where does it truly begin or end? What does it mean to age, to grow, or to exist, when even the smallest organisms can step outside the flow of time? Even within limits, tardigrades stand as sentinels at the edges of possibility. They do not defy death. They reveal its shape, narrow, silent, patient. In the quiet of moss, the stillness of microscopic soil films, they show us the subtle, almost invisible boundaries of life itself. Across Earth, countless other organisms have evolved to persist where life seems impossible. From deep-sea vent worms to high-altitude insects, the rules of biology are stretched in ways we are only beginning to understand. In this series, we explore these margins, where evolution tests the limits of survival and where life continually surprises.